On this week's Arabidopsis Research Roundup, I'm again delighted to welcome Lars Ostergaard from the John Innes Centre to talk about a new paper just published in Plant Cell, the title of which is Auxin-Induced Modulation of Etten Activity Orchestrates Gene Expression in Arabidopsis. So this is a follow-up paper to a paper we discussed at the tail end of 2016, and you can listen to that on the GARNet YouTube channel. I'll put a link associated with this. So basically in that paper, Lars showed that his group, in collaboration with European collaborators, put together or defined a non-canonical auxin signaling mechanism which revolved around the Etten auxin response factor. So thank you, Lars, for joining us again. It would be great to hear now how you've taken that work on and looked more specifically maybe at the role of Etten in this process.
Thank you very much, Geraint. And yeah, thanks for giving me this opportunity to provide a follow-up review of the next paper here. So yeah, I mean, one thing that we found out, as you rightly said was that Etten interacts with a range of very different transcription factors to regulate gene expression, and that these interactions are affected by auxin in some ways. And that's what we wanted to find out in this follow-on paper was really at the genome-wide level. How significant is this? In the original paper, we described a specific gene, pinoid, which regulates auxin transport or direction of auxin transport. But we wanted to find out now, at a more genome-wide level, how Etten does this, with which factors, and what are the genes that are sensitive to this non-canonical auxin signaling pathways, and which are not, but are still Etten targets. So we took this ChIP-seq and RNA-seq approaches, so treating Arabidopsis inflorescence tissues with and without the naturally occurring auxin indole acetic acid, or IAA. And we also, this was for a ChIP-seq approach where we used a reporter line where we had a TFP attached to the Etten protein. And then we also did an RNA-seq approach in the wild type versus the Etten mutant background. And in this way, we found different sets of genes. Genes where Etten binding to their promoters or intronic sequences was dependent on IAA or independent of IAA. And also genes whose expression was dependent on Etten, whether IAA was present or not. And this gave rise to this perhaps a bit complicated, although we did everything we could to make it as clear as possible. In one of the figures, a schematic that really shows these different sets of genes and how they are affected by Etten and auxin in combination. So from those, we were able to go in and specifically study how some of them were regulated in more details. Both which part of the genes were bound and was significant. Also, we tried to make comparisons to other transcription factor binding sites that were already published, such as the repromless chip that was published by Robert Zablowski's group recently, and found that there was some overlap in gene targets between Etten and repromless. And sometimes the binding of Etten to these genes was indeed dependent on repromlase. At least in one case, we found that was true. We found in this paper here also a number of specific groups of genes. For instance, some that were involved in hormone dynamics. And specifically, we found that Etten seems to be directly binding genes from all levels of auxin dynamics, from biosynthesis, transport, and signaling, and very often act as a repressor of those genes. So that gives a nice sort of feedback loop. Etten is both affected by auxin itself, but it also regulates the genes that are mediating auxin dynamics. We also targeted in on some genes that are involved that are belonging to the class of basic helix loop helix type transcription factors. We knew from the work that we published in this development last year that Etten interacts with one of those called the indehiscent, and that interaction, protein-protein interaction, is indeed sensitive to IAA. Now we also found 4, and specifically characterized 4 genes belonging to this family as well, And in one of these cases, Etten also interacts at the protein level. So it both regulates its expression, but also interacts at the protein level, which is in an IAA-sensitive manner to regulate gene expression. So again, providing a feedback mechanism for how Etten regulates its downstream targets.
Okay. No, I was going to say, Lars, you said that perfectly, but it really strikes me, this paper, there's two parts. There's this large genomic part, and then you do zoom in then on specific genes with which Etten interacts. So I wanted to quickly ask a question about the kind of genomic aspects of it. So you said you have an expression, RNA-seq, and also a chip-seq portion of it. A lot of the times it looks as though the Etten is binding, as I say, in promoter regions or in intronic regions. Do you see any correlation between when there are genes which are highly expressed in the wild type versus the Etten, whether they bind a specific part of the gene? Are they upstream? Are they in the intron? Were you able to get to that level of detail?
No, we haven't actually. We have not been able to find a correlation with that. Okay. We did look into it, absolutely, and but we have not been able to find any correlation about where binding is compared to how the expression level is. Okay. I mean, one thing which is fantastic in these days is, of course, that all these data are now available out there for people to do also their own analysis of these data. And it might be that we have not done the right analysis, and there might be some correlations out there, as you say, that we didn't pick up. Yeah. But from our analysis, we did not see this.
So as you said, towards the end, so you have a model with basically different modes of interaction dependent on direct targeting and different concentrations of IAA. So can you fill us in on what you're able to conclude from that?
Yes, that's a very good point. So at the end, indeed, the model suggests 4 different scenarios where different transcriptomes are being activated or only deep repressed. So we start out with a transcriptome we call A, which could, for instance, be Etten alone, either repressing or activating a certain set of genes. Yeah. And these genes are seemed, at least from the classes that we have characterized here, to very often be hormone genes that are involved in some kind of hormone dynamics. Then in other situations, we believe that Etten works certainly with other transcription factors that we here call X, and then they regulate a different transcriptome that we call transcriptome B, here with a different set of targets. And some of those here, we think that X is definitely can be repromless. This is what we characterized in this paper. Here we also previously shown that indehiscent can be one of those as well, and I'm sure there are many more. We did identify several more that are also IAAs, where the interaction with Etten is IAA sensitive in the gene development paper. Then when Auxin becomes present, then we believe that IAA has an effect on the interaction between Etten and the other transcription factors. What exactly the molecular nature of that effect is, we're still working on that. We don't know yet.
Right.
But then we believe that perhaps in other cases we have a situation as in for the transcriptome D, where the interaction of Etten with the target genes requires the binding of a different transcription factor, such as is the case for one of the repromless targets. And there could be others like that as well, where the transcription factor provides a kind of an anchor point for Etten to enter and bind in case, for instance, the interaction of Etten with this promoter sequence is not strong enough. Okay. So these are the kind of scenarios that we can envision based on our genome-wide data set that can take place the way that Etten regulates its target genes.
Yeah, absolutely. So I think in the paper you mentioned still that maybe the tier 1 based paradigm of auxin signaling might be the main one, but I would argue that there might be more information. We know more information about how Etten works now, specifically the Etten protein compared to any other AuxIA protein or any other ARF, really, with the level of detail which is provided within this paper, I think.
I agree. Thank you for pointing that out.
I think you would agree, though. Yes, that's true. That's true. No, and I think the beauty of science, right, and the beauty of this paper really strikes home again. That the more you know, the more you need to know, right? You find that there's interactions, but then there are so many interactions which Etten doesn't interact with replumnus, so there must be something else involved, and we have no idea what that is at the moment. So,
yes,
and I'm sure
right.
That'll something you'll be following up on.
Yes, that's exactly right, Lorraine. We would. This provides sort of a platform for which we can now go and find out much more about the regulation in much more molecular detail, much deeper molecular detail.
Excellent. Okay, so thank you again very much, Lars, for this. So we've had an update 9 months on, a gestation period for this Etten paper, and we'll see where we go maybe in the next couple of years, and hopefully we'll be able to check back in with you at that time again.
That would be fantastic.
All right. Great. Thanks very much.
Thank you, Greg.